To figure out how we inherit hereditary characteristics from our parents and ancestors, we must know about the great work done by scientist Johann Gregor Mendel. In this video, we will study about Mendel's monohybrid and dihybrid cross. Before we study about Mendel's monohybrid and dihybrid cross, we need to know certain terms, the first ones being dominant and recessive characteristics. Now, individuals or babies or offspring or progeny receive two versions of each gene from each parent. If the genes are different, then only one characteristic will be expressed or shown in the baby, and that will be the dominant one or the dominant gene. The effect of the other gene from the parent is masked, not shown, that is called recessive. For example, a child has parents having one with curly hair and one with straight hair. Now, genes of both curly hair and straight hair are present within the progeny or the baby. Now, that baby may show curly hair, meaning curly is the dominant gene. It also has the gene for straight hair, but it is not shown because it cannot show curly hair and straight hair together. Therefore, one gene which is shown from the one parent is the dominant one, and the one which is not shown but there present within the progeny is known as the recessive gene. Secondly, homozygous versus heterozygous. Homozygous is a condition where an individual, now we are talking about the baby or the progeny, inherits the same genes. Maybe both parents have curly hair. Okay, so it will inherit the same genes from both parents. That condition is known as homozygous. And heterozygous refers to having inherited different forms of the particular gene from each parent. Genotype versus phenotype. Now, a genotype is an organism's set of inheritable genes that has been passed down from parents to offspring. Whereas phenotype refers to the physical properties like appearance, development, and behavior. What I can see in the baby is his phenotype, and what set of genes he has inherited is his genotype. Mendel's principle of heredity. His principles are based on the fact that genetic material or genes is transferred in equal quantity from each parent to the baby or the progeny. Gregor Johann Mendel, he was an Austrian scientist. He carried out research and put forth certain principles of heredity. And all his experiments were conducted on what he could see in the pea plant, that is Pisum sativum. So you can see here, he is showing you the dominant and recessive characteristics. The shape of the seed, dominant characteristic is round and recessive characteristic is wrinkled. Color of the seed, again we had yellow color which was dominant, green which was recessive. If we come to the color of the pod, we see green is dominant, yellow recessive. Okay, position of the flower, axillary position, that is somewhere in between near the nodes, and terminal position, that is right on top. So the recessive characteristics were the terminal position. Mendel's monohybrid cross. Now, a cross between two pea plants where only one pair of contrasting characters is taken is called a monohybrid cross. Now, previously we seen that there was shape of the seed, color of the seed, height of the plant, color of the pod. Now, when we take only one pair of contrasting characteristics, for example, only the color of the seed, and here we are taking only the height of the plant, whether it's a tall plant or a short or dwarf plant. So, Mendel used tall and dwarf pea plants. Now, in tall shown by capital TT, showing that they are dominant characteristics, and when we use lowercase characters, alphabets like small tt, it shows that those are recessive characteristics. During gametogenesis, these genes separate from each other and the gametes T are formed. See that this is the parent T and T, recessive dominant and recessive short characteristics. Tall plant, dwarf plant, they are the parents. Both of them have two gametes, that is capital TT and small tt. Now, when the gametogenesis, joining of the gametes, take place, they separate, okay, into single and single. Then they come together, and in the first generation, you get a mixed capital T and a small t. Okay. All plants in the F1 generation, that is the next generation, were tall, but they contain the factor for dwarfness too. That is, their phenotype, how they looked like, was tall, but their genotype, what they are made up of, was mixed. So once again, a tall plant shown by capital T and T, a short plant or dwarf plant shown by small tt. During gametogenesis, the gametes, they separate, okay, then they come together in the cross, and they are showing that they are tall because the T or capital T is the dominant characteristic. So in the F1 or the first filial generation, all plants were mixed like this. They got equal number of tall and dwarf characteristics, but all of them were tall because the characteristic of being tall is dominant over recessive. Okay. Mendel continued the experiment and then again self-fertilized the F1 plants, and we got another filial generation, that was the F2 generation. In the F2 generation, both tall and dwarf plants, phenotypic ratio was 3 tall to 1 dwarf. The genotypic ratio was 1 fully tall, 2 which were mixed genes, and 1 fully dwarf was shown in the Punnett Square. Okay. Now let us see right from the beginning what happened. We had a tall plant having the gametes T and T. We had the dwarf or short plant having the gametes small t and t. During gametogenesis, they separated, okay, and when the plant was in the F1 generation, that is the first filial generation, we got plants with mixed genes, okay, capital T, small t, but the plants looked tall because they had the dominant characteristic capital T, that is showing that they are tall. Then on self-fertilizing these mixed plants now, can you see a mixed ones, TT, TT. Again you will find that they separate during gametogenesis, TT, TT. Now we will write this in the Punnett Square. This is called a checkerboard or a Punnett Square, where the male characteristics are written, male gametes are written here and the female gametes here. Can you see male gametes T and T, female gametes T and T. Now we'll cross these two. T into capital T will give you this, T into this giving you this. Then we go to from here to here, which will give me a mixed one, and here to here, which will give me a this. Now, when both are the same genes, we call them homozygous, both tall. Then we have mixed genes, so heterozygous or mixed gametes, but they will be tall because there is one dominant characteristic here, again another heterozygous, and then both same, therefore it will be homozygous but dwarf plant. Therefore, when I look at it, I will find my phenotypic ratio 1, 2 and 3 tall, whereas only one short. This is the phenotypic ratio. Whereas my genotypic ratio, what it is actually made up of, is only one homozygous tall, 2 mixed and 1 short. So the genotypic ratio will be 1:2:1. Coming to the dihybrid. Now, a cross where two pairs of contrasting characters taken is called a dihybrid. Now, the two pair of contrasting characters taken, that is yellow dominant versus green recessive, and round will be the dominant versus the wrinkled seed, which is the recessive. Now, the P1 generation or the parental generation consisted of round yellow seeds, dominant, and green wrinkled seeds, which were. During gamete formation, the gametes separate and they formed RR and RR gamete. Now, in the F1 generation, as expected, plants produced yellow rounded. Though the genotype of these plants were mixed, that is, they had all characteristics, yellow, green, round and wrinkled together, but they looked like their parents, yellow and rounded. Okay. Plants of the F1 or the first filial generation. After the parental generation comes the first filial or F1 generation. Now they produced 4 types of gametes, mixed gametes. One was capital RR, RY, that is for round and yellow. Then we had this one, round and green. Then we had wrinkled and yellow. And this one totally recessive, wrinkled and green. And of all these 4, only the first one, that is RY, and the RY, which is the lowercase alphabets, dominant and recessive, was similar to the parents. The F2 generation, next generation, after selfing or self-fertilizing the F1 plants which we got, the mixed plants, 16 different possible combinations were got, okay, by the union of 4 male and 4 different. These combinations are shown in the Punnett Square. Okay, Punnett Square. Okay, on the top we have the separated male gametes during gametogenesis. Okay, and we have on the left side the female gametes. Okay, I cross this and this, I will get this RR dominant characteristics. Then I go to the next, where I will get capital R's, 1 Y and 1 small y. Okay, similarly I will start with this one here and I'll get this answer. Finally I'll come with this into this will give me this into the last one will give me this, and so joining or crossing the male gametes and the female gametes, we get 16 different combinations. Now we know that capital Y and capital R stands for round yellow seeds, whereas lowercase or small r and small y stand for wrinkled and green seeds. Okay, green seeds. So now, by looking at these 16 different combinations, phenotypic ratio, just by looking at the plants, of 9 showing round and yellow seeds, 3 showing round and green seeds, 3 showing wrinkled and yellow seeds, and only 1 showing totally recessive, wrinkled and green seeds. So let us check now. We have all mixed type of things here, but you also know there are dominant and recessive characters. The dominant ones are shown by the capital letters. And even if they are mixed, but there are the capital letters here, we will see the seeds as round and yellow. So let us check for these 9. First one is here, round and yellow. We need to have only one gamete, round and yellow, capital letters, to show us that the seed is round and yellow. 2, 3, 4, 5. This one is not there. 6, 7, 8. Not here, not here. 9. Not here, not here, and not in the last. So we have 9 showing that they are round and yellow. Now let us see for this one. Round and green. Round and green, this one here. Round and green, 1. Not here. Round and green, 2. And round and green, 3. Now we have wrinkled and yellow. Wrinkled and yellow, 1 here, 2 here, and 3 here. And finally, the one which is totally green and wrinkled, only 1. Now, this is how plants look like. But you know, genotypic ratio is what they are made up of. And if you look, everything is so mixed up. So we have one which is totally, dominant. Then different types of mixtures over here. And if you look and start counting, you will have the genotypic ratio 1 is to 1 is to 2 is to 2 is to 4 is to 2 is to 2 is to 1 is to 1. So that's all about the monohybrid and the dihybrid cross by very renowned scientist Gregor Johann Mendel.